I want to say just a few more things about hydrogen bonding. Here are some potential energy surfaces for hydrogen bonding where the y-axis represents energy and the x-axis sort of represents the position of the hydrogen in between donor and acceptor. And you can see in panel A, this is a situation where it's more favorable for the hydrogen, for the proton to be on the hydrogen bond donor than it is for the hydrogen to be on the hydrogen bond acceptor. But going from here to here represents a proton transfer, okay? And you can sort of think of the relative energy levels of these two situations as being more or less defined by the pKa values of the acids in either well, okay? So this would be a case where when the proton is on the donor, it's more stable, whereas when the proton is on the acceptor, it's less stable. So this one would have the lower pKa because it'd be more acidic. This would have the higher pKa because it's less acidic. And you can see there's a barrier to interconversion. And these lines represent allowed energy levels, allowed vibrational energy levels within these energy wells. Notice that you can never get to the very bottom of the well because the energy levels are quantized and you still have what's called 0 point energy at the lowest vibrational level. It's thought that if you have donor and acceptor atoms where the acids on either side have similar pKa's, that the barrier for interconversion gets lower. And you can imagine this vibrational state at this lowest line tunneling through the barrier and sort of occupying all of that space. And of course the limit would be either end is so similar in stability and they are so close to each other that there's really no barrier at all. It's sort of controversial. You'll hear people sometimes talk about low barrier hydrogen bonds. And these are supposed to be very stable. And some people have attributed the ability for enzymes to catalyze certain reactions as being related to the formation of a low barrier hydrogen bond. If you remember learning in biochem about serine proteases, you remember the catalytic triad, there's an aspartate and a histidine. Some people have hypothesized that the aspartate and the histidine form a low barrier hydrogen bond that pays for the serine nucleophile to attack the carbonyl carbon. The issue is somewhat controversial. The ability to form a low barrier hydrogen bond is highly distance dependent and also dependent upon the pKa's of the amino acids. So it's sort of an issue that hasn't gained a huge amount of currency, but the principle that if you have a hydrogen bond donor and an acceptor and the pKa's of the corresponding acids are matched, you can have a pretty stable hydrogen bond. The other thing I wanted to say about hydrogen bonding is your book chooses this example, and I have to mention it because it's from my PhD advisor's lab, though it's really not hydrogen bonding that holds this thing together. So this is a beta peptide. Years ago, Professor Gelman asked the question, could we somehow make unnatural molecules that would adopt the kinds of regular shapes that proteins adopt? And this is sort of a fundamental organic chemistry question. Why does nature choose alpha amino acids where there's only one carbon between carbonyl carbon and nitrogen? Why couldn't there be more? And so he explored making peptides with beta amino acids. Just to orient you, if you've not done a lot of amino acid or peptide chemistry before: carbonyl carbon, alpha carbon, beta carbon, and amide nitrogen. And it was known at the time that you could make, if you made a beta peptide sufficiently long enough and with side chains at the beta carbon, you could get them to form helices, though you needed to have the beta amino acid be kind of long, and you needed to do things like arrange salt bridges to make it happen. Professor Gelman's chief insight was that if you used a cyclohexane ring, you could pre-organize this amino acid in a conformation that would be maybe consistent with helix formation. Just to be clear, an alpha amino acid looks like this, and there are two dihedral angles to worry about. You've got the phi dihedral angle and the psi dihedral angle. In beta amino acids, you have an additional dihedral angle to worry about. There's 3 rotatable bonds. And so it's thought that by incorporating another group in the backbone, you maybe make it more entropically costly to form a helix because you've got more things to constrain. And Professor Gelman's insight was, why don't we constrain the alpha and beta carbons to be part of a ring? I don't actually know the story, whether they made models first and played around with them or whether they just empirically tried. They chose the trans relationship between the amide nitrogen and the carbonyl carbon. And when they made this, they found, and this is only, let's count, 1, 2, 3, 4, 5, 6 amino acids. Remember what I told you last time, alpha helices don't generally form for peptides that are that short because the cost of nucleating the helix is too entropically unfavorable to be paid off by the enthalpy you get from making hydrogen bonds. In this case, by incorporating the 6-membered rings, you have already paid a lot of that nucleation cost. And so these things fold right up at this short oligomer length. And shown here is the kind of helix that they form. So let's see. Here's the N-terminus. Here's the lysine side chain, beta carbon, alpha carbon, carbonyl carbon. Here's a 6-membered ring. And you can see how the 6-membered ring sort of puts the amide nitrogen and the carbonyl carbon in a fixed sort of twisted relationship, which generates the helical turn. And if you follow the ribbon around, you can see that this forms a helix. It turns out that if you look down the helical axis, one of the things you observe is that the side chains sort of repeat come back to the same position around the helix in sort of a 3 residue repeat. So every 3 residues you're back on the same side. And that's why, for example, this cyclohexane residue, number 2, is on the same side of the molecule 3, 4, 5 as cyclohexane number 5. And the Gelman group has gone on to use this regular repeating pattern to design beta peptides that have interesting functions. They've used them to inhibit antimicrobial activity by making one side of the helix positively charged and the other side of the helix greasy. And they've worked on generating molecules like this that can inhibit protein-protein interactions. And they found that you don't actually need it to be all beta amino acids. You can mix and match alpha and beta, and they've done some really cool stuff with that. Your text uses this to demonstrate that indeed there are hydrogen bonds in this beta peptide helix. Maybe not a surprise there. The one thing we could mention is, and I think for comparison, I probably am going to need to show you a helix. No? sorry, let me just copy something that has a helix for comparison. Copy. This is useful because it'll show you the difference between a beta peptide helix and a peptide helix made of alpha amino acids. If you look at this helix, this is the N-terminal side of the molecule. This is the C-terminus. All the nitrogen-hydrogen bonds are oriented in this direction, and from the N-terminus toward the C-terminus. Right? Nitrogen's more electronegative than hydrogen. Similarly, all the carbonyl oxygens are pointing towards the C-terminus. And so you, in an alpha helix, you have this alignment of each of these sort of local dipoles, and that generates an overall thing that we call a helical macrodipole. And this can have some important consequences in protein folding. What it means is the N-terminus of an alpha helix tends to be electron poor, the C-terminus tends to be electron rich. If you compare that with the beta peptide helix, this is the N-terminus, this is the C-terminus. Now the nitrogen-hydrogen bonds are arranged in a different direction. The nitrogens are towards the N-terminal side of the helix. The carbonyl oxygens are also oriented towards the N-terminal side. And so what this means is this beta peptide helix has a reverse helical macrodipole relative to the alpha helix. And you can ask whether or not that would have some interesting consequences. All right. Yes?
If you put a bunch of these beta amino acids together with tRNA ribosomes, are they compatible?
That's a fantastic question. And the Gelman group has hoped for years that someone would figure out how to make the ribosome accept beta amino acids. And so far, no good. It turns out the ribosome can accept different side chains, and there are a lot of people who've worked on that. There's many strategies for that, but it doesn't appear to like that extra methylene group for reasons that aren't immediately clear. So,
is the ribosome itself or the tRNA?
That's a great question. Is it the ribosome itself or is it the tRNA? Having a tRNA, amino acyl tRNA synthetase that charges a cognate tRNA is itself a difficult problem. But you can synthesize a tRNA and put the amino acid on it yourself, I think, maybe. So if you could do that, the question would be, would it get incorporated? And I think the answer is still no. So these are made synthetically. And of course, if you wanted to access larger types of molecules, you could do something called native chemical ligation, which is a way of stitching together individual subunits in a longer chain. Yeah?
Yes. Someone using unnatural amino acids, do you have to engineer a new genetic code for the ribosome to use?
If you were going to use unnatural amino acids, would you have to engineer a new genetic code? It depends, but there's a lot of work by people like Floyd Romsberg and to some extent Eric Kuhl. Romsberg's at Scripps, Kuhl is at Stanford, and others who have been trying to expand the genetic code by actually coming up with unnatural DNA base pairs with the idea that someday if you had a larger genetic code you could incorporate other things. It's still kind of a pipe dream, although Romsberg's work has gotten pretty close has definitely succeeded at some limited genetic code reprogramming and incorporation of unnatural things. If you want to read more about this area, you can go to this classic paper and accounts of chemical research, 1998. The title of this is Foldamers, a Manifesto. Sam Gelman has kind of a flair for writing, and so that's sort of where he introduces the term foldamers, which he coined and then has been widely adopted.
Also, with the rings and all being stacked like that, it makes the long range structure look like cellulose, except without all the hydrox.
Yeah, well, yeah, you got the 6-member ring that sort of seems to resemble a carbohydrate. The field of foldamers has really exploded. There's a lot of people doing a lot of different things with unnatural molecules that can nonetheless adopt regular shapes. And one question you have to ask is, what? So what? What good is making that, aside from the fact that you can make it? This is where science becomes gee whizology. And usually the first time you make something cool, everybody's like, ooh. But after a while, people get kind of like, bored. So you have to do things useful with these molecules. And so Sam's funded his research for 20 years or more based on the idea that we want to use these molecules to do important biological functions, and perhaps even functions that biology can't do because biology isn't using this alphabet. Anyway, let's see. So hydrogen bonding. The next area I think is kind of fun. We'll call this interactions involving pi systems. And there's a lot of different flavors here. The key principle that explains what's going to happen here is that benzene is a nonpolar solvent. It's not particularly miscible with water. However, it's not a simple greasy hydrocarbon because it doesn't have a uniform distribution of electron density. So in terms of electron density distribution, we've talked about dipoles before, right? Dipoles represent a linear distribution of electron density. And you, well, I don't want to necessarily confuse the issue. A quadrupole is a non-uniform distribution of electron density in a molecule. And there are specific characteristics of a quadrupole. Your text notes that dipoles tend to resemble p orbitals in the sense that you've got one side and the other side that are in phase, not charge. That is maybe a little too far, but it may be a reasonable analogy. Your text points out that quadrupoles tend to look like d orbitals. So here's the dz squared. I only know that that's what the dz squared looks like because I looked it up. I don't think about d orbitals a lot. Shame on me. And so a quadrupole is going to have a positive end or here and then a negative end surrounding it, or vice versa. So if we look at benzene, you know that sp2 hybridized carbons are more electronegative than sp3 hybridized carbons because they're increased s character.
If we continue on then, does this mean that octopoles will look like f orbitals?
I think so, but I don't even know what f orbitals look like. So again, shame on me. And I don't have any good examples of what octopoles are. Other than if they lose a pole, do we call it a septipole? Like Hank the Septipus from Finding Dory? No? Nobody knows that one? Okay. All right. So imagine sp2 hybridized carbons are more electronegative than hydrogens. And what that means is that the hydrogens around the ring are electron poor, and the carbons within the ring are electron rich. And if we were to look at the ring from the side, and sort of plot the areas of positive electrostatic potential versus negative electrostatic potential, again, you're going to see the edge of the ring as electron poor, and then you're going to have areas above and below the ring that are electron rich, sort of like Saturn. All right? So, with that in mind, you can now predict some things about how other molecules might interact with benzene. So, suppose let's just actually talk about benzene's interaction with itself. Okay? Yeah.
This also explains the NMR shift of benzene then? The hydrogens are outside?
Does it explain the NMR shift of benzene? In a sense, yes, but it's more than just electron poor. In benzene, you have ring current that actually reinforces the external magnetic field. And those two aren't necessarily tied together. Yeah, but they sort of coincide. So, what do you think about benzene interacting with itself? I will redraw benzene on its side, and then I will copy and paste to get another benzene, and then we'll do some low-tech animation to get an idea of how benzene molecules might interact with themselves. So, here we go. What do you think about a direct parallel stack? What's wrong with that? Repulsion, right? You're bringing the electron poor portions together and the electron rich portions together. Okay, so yes, in fact, in benzene, we do not see, either in the gas phase or in solution or in various crystal structures, we generally do not see benzene do the direct above and below stack. All right? How about slightly offset? That's going to bring the electron poor portion of one ring into the area above the electron rich portion of another ring, and vice versa. All right. So this is often called offset stacked. All right. So sometimes we tend to know, I'm not sure what your text calls it, no direct overhead stack. I'm not sure. Sometimes we draw a conclusion based on DNA structure where there is a lot of overhead stacking, and we apply it to benzene and think, oh, that ought to work just fine. It actually doesn't. The electrostatics, the quadrupole doesn't support that. It's got to be offset. Now you can ask, why does it work in DNA? Well, DNA has a bunch of heteroatoms in the ring, and that changes the electronics of the ring and allows them to be more compatible with each other. So overhead stacking is a thing in DNA, but it's not a thing for benzene. There's another alternative here. Any other thoughts as to what might work? I gotta try to erase stuff, and then I gotta try to see if I can get one benzene molecule away from the other. Any other ideas? Go ahead. What? T-stacking. What do you mean by that? So two of them perpendicular. Okay. So, whoa, yeah. So what about that, where, yeah, you have the electron poor part of a benzene stacking into the electron rich face of another one. Yep, that's called T-stacking, and it's actually a little bit well, it's not controversial, but there's a couple of different options that you tend to see. I think they're both probably called T stacking, but it's an open question as to whether you need just the one proton hovering over the ring, or whether you can tilt this a little bit so that two protons are pointing at the ring. And you can actually calculate the energies of these various benzene dimers in the gas phase, and they range from 2 to 3 to 5-ish kilocalories per mole, depending on how you do the calculation. And you do see these kinds of interactions in protein structures. Of course, in proteins you have aromatic side chains, and there are lots of examples of this kind of interaction. I can show you one of them, maybe. No? Well, let's look up the villain headpiece domain. This is a part of a chicken protein called villain, and this is the, not like as in bad guy, but and this is the headpiece of that domain. And I'm going to try to get to a structure RCSB and get the structural database. Crystal structure of villain headpiece. I kind of want to see the side chains. Polymer. Cartoon. Dang it. Anybody know how to use RCSB? Oh, can I can, but oh, right. Oh, actually, maybe IMOL view will work still. It's no longer supported. 2RJY. 2RJY. I don't know how to use PyMol on my laptop. It's irritating. I want to see, no, that's too much. That'll work. Okay. So this is the inside of the villain headpiece domain. It's held together by interactions among 3 different phenylalanine residues. You can see the benzene rings sort of there. And if you were to make space-filling versions of those benzene rings, you'd see some either offset stacking, yeah, you looking at these two, there's sort of one sort of hovering over the other, and it's a little bit tilted. Some of the tilting has to do with the geometry of the molecule. I wonder if I can make space filling of just a selected few. Well, I won't worry about that. A guy who was in the Gelman group, he's actually a BYU graduate, his name's Matt Wall. He did a project in the Gelman group on this protein, and he demonstrated that if you replaced some of these phenylalanines with pentafluorophenylalanines, the domain got stronger. And that can lead us to another discussion. Supposing you wanted to do the overhead stack thing, can you think of something you could do to one of the 2 benzene rings to maybe make overhead stacking more favorable? What? Make one of the rings electron poor. Okay, how do you do that? Add withdrawing groups, and fluorine is often our electron withdrawing group of choice because fluorine's so electronegative and because it's also small. And so if you have hexafluorobenzene and then just regular benzene, only in that case is the overhead stack is okay. And this is because the electron-rich face of one of these rings interacts with the electron-poor face of the other, and the electron-poor edge of one ring interacts with the electron-rich edge of the other. So they're very nicely matched. And there are a lot of people who've looked into incorporating fluorinated side chains into protein structure and enhancing folding. If you want to know more about that, you could look up the villain headpiece papers by Wall and Gelman in the early 2000s, sort of looking around 2003. Also, Professor Jianmin Gao at Boston College has done a lot in recent years with putting fluorinated amino acids in protein cores and seeing what they do. Okay, so those are the so-called pi stacking interactions. I didn't actually intend to talk about them first, but there you go. What else do you envision being able to interact with a benzene ring? Ions. Okay, great. So suppose you're a positively charged ion, and here is the benzene ring that is sort of sad and lonely. Maybe there, looks a little sadder. If you're a cation, where do you think you're going to be? Right. Some cation, we'll say, I don't know, sodium, could actually hover right over the ring. And sometimes people will draw a dotted line to indicate that the cation is interacting. And often it will be within van der Waals distance of the ring, like it's sort of sitting down on the ring. So I'm going to paste in a figure from your text that talks about these things. So in the top panel is shown a cation at this particular distance hovering over a benzene ring. And then over here are free energies of complexation, how much more stable the complex is versus the ion and the benzene on its own. These can be measured, I think, using mass spectrometry, but I think they can also, I think this is from Doherty's work, and these I think are calculated. The lithium interaction is stronger. Why do you suppose the lithium interaction is stronger than the sodium interaction? Lithium's smaller, so maybe it can get closer. Also, in lithium, because it's smaller, the charge is all sort of tighter in one place and less diffuse. And so, yeah, it follows atomic radius trends as you go sodium, potassium, rubidium, and so on. Interestingly, and Doherty was here a couple years ago and gave a talk, and he pointed out that potassium interacts with benzene in the gas phase just as strongly as it interacts with water in the gas phase. So what that tells you is that these interactions, even though you think of benzene as being nonpolar, you have to remember its quadrupolar character, because that is involved in a lot of its function. Okay, and let's see. So almost immediately, as one of the things you think about almost immediately when you're dealing with interactions involving a benzene ring is, let's add groups to the benzene ring and see what happens. The reason we like to do that, I think, is as organic chemists, some of the earliest cool reactions from like the late 1800s were electrophilic aromatic substitution reactions. And that was, in many cases, our first exposure to the idea of a planned out synthesis in sophomore organic. So we're really kind of excited about, I want to put phenol, or I want to put some other electron withdrawing group. I want to try chloro versus bromo. So here are some binding energies between sodium and substituted benzenes. And the trends are kind of interesting. Comparing benzene to fluorobenzene, the interaction energy goes down. Comparing benzene to cyanobenzene, it goes down quite a bit. It doesn't change very much on adding an OH group, and it gets actually a little bit better on adding the amino group. All right, so let's try to rationalize these trends. What's the first thing that comes to mind? Donor versus withdrawer. Okay. So when we thought about electrophilic aromatic substitution, we identified groups that were resonance donors versus resonance withdrawers. We talked about polarizing the pi system of the ring to direct the regioselectivity. Okay. Fluoro is a resonance donor but a electron withdrawer because of its electronegativity. Does that explain why, justify why the fluorobenzene binds sodium less? Okay. Right. It's all about whether or not the ring is electron rich. You're used to thinking of oxygen as a donor, a resonance donor, and indeed it is an ortho and para director. However, it binds maybe a little bit less goodly well. Binds a little less well to sodium than benzene does. What's going on there?
Have you used in the gas phase or is this inside?
Gas phase.
Gas phase.
What do you think? What's going on there?
I was speculating maybe there's competition for the sodium to bind to the benzene versus the hydroxyl group.
Okay, so you can think of some coordination, alternative coordination with the hydroxyl group. Is that competing with the benzene ring? I don't actually know, though you could calculate what those relative energies are. So that'd be interesting, yeah.
The OH is electron donor by resonance, but shouldn't it be withdrawn by inductive effect?
Yeah.
Oxygen is electronegative.
It's an inductive withdrawer. It is a resonance donor. So this may be a clue that inductive withdrawal is perhaps more important than resonance donation. Now, nitrogen binds the aniline, the aminobenzene, binds sodium better, and you could rationalize that by saying, well, even nitrogen's a little bit less electronegative than oxygen, and it's a better donor as a result. So it turns out Doherty and others argue that the right way to think about substituted benzenes and cation pi interactions is to focus mostly on the inductive characteristics of a group rather than its resonance characteristics. And in fact, what Doherty suggests is that instead of looking at the ring and looking at these structures and try to rationalize it, you just calculate the electrostatic potential surface, and then you can rationalize the results that way. So I'm going to rely on Google again. I probably should have prepared these figures. ESPN, no. Electrostatic potential surfaces substituted, misspelling it. No, that's pretty good. Benzenes. Okay, great. We'll choose a few of these. I don't know which to choose now. Okay, that's good. There we go. That's sort of what we want. No, come on. There we go. All right. I don't know what fictitious means there, but if you look, for example, at benzene, you see red here is electron density, is negative electrostatic potential, and then blue is positive electrostatic potential. What you do to calculate this is you basically say, okay, I'm going to put a test point charge anywhere along this surface of the benzene molecule, and I'm going to measure the interaction of that positive test charge with benzene at that surface. And so when it's red, that means electron rich, blue means electron poor. And I don't know. This is a computational paper, and I'm not sure what it means. But notice nitrobenzene has, whereas benzene is red, nitrobenzene is green, which is basically sort of neutral. And I want to show you the difference between benzene and phenol. So that's, maybe I'll just put phenol in the search terms and see what we can come up with. Oh well, maybe I'll show you some of that another time. Basically, if you compare benzene with phenol, you see that the electrostatic potential surface, and these are actually in your text, so you can go look at those at the back, but benzene's sort of bright red here, or redder, and phenol is sort of orange/yellow in the middle. So in terms of the actual electrostatic potential of the surface, phenol's not as electron rich in the ring as is benzene. There's more. There's some more controversy here. I'm going to just provide a reference that you can go look at if you want to. This is actually from Dan S.'s PhD advisor. And what they demonstrated is that in most cases, you can rationalize, they did this through calculations, you can rationalize the differences in binding energy between, say, sodium and these various monosubstituted benzenes, not by considering polarization of the pi system, but just by considering the through space interaction of the sodium ion with the substituent itself. There were exceptions to this observation for the most vigorous sort of resonance electron donors like the aniline, but for the most part, it was like you could really predict what the binding of sodium to benzene would be by considering the binding of sodium to benzene and then adding on what you would predict for binding of sodium to whatever the group was there at that distance. So, moral of the story is cation pi interactions track with the electrostatic potential of the ring and not necessarily with what you think is a good electron donor from your electrophilic aromatic substitution intuition. All right. Sorry, I feel like I muddied that a little bit. Any questions you want to ask? Any confusion that I've created instead of cleared up? Maybe we'll look at some cation pi interactions. So this is from a protein, and I forget what protein it is, but the PDB code is there, and we can look it up. So in blue, we have residues, amino acid side chains on this protein that are positively charged. Lysine is an ammonium side chain. Arginine is a guanidinium side chain that's also positively charged. This is another arginine. This is another lysine. And you've got this extended array of positive charge on one side of an aromatic ring. This happens to be tryptophan, sandwiched with another positive charge, sandwiched with another aromatic ring, another positive charge, another aromatic ring. And these types of interactions are actually all over in proteins, and there's a lot of bioinformatics studies that talks about how frequent they are in proteins and whether they tend to be more with just the benzene ring, which is phenylalanine, or whether tyrosine or tryptophan is better. Tryptophan, actually, if you look at the indol's electrostatic potential surface, it's way more electron rich than benzene is, and so you'd predict that those interactions would be more stabilizing. One interesting thing to look at here is if you think about an ammonium group, we would normally draw the positive formal charge on the nitrogen, right? Go ahead.
Also, does histidine engage in these kinds of interactions because it has an aromatic ring?
Histidine can. It has an aromatic ring, and it also could be a cation. So there are examples of histidine interacting with benzene rings, for sure. So normally we would draw the ammonium with a positive charge on the nitrogen, but if you look at the group that is closest to the aromatic ring, it's actually this carbon, the epsilon carbon of the lysine side chain. What do you suppose is going on there? Yeah, Kim, go ahead.
Well, nitrogen is more electronegative than carbon, and so it's starved for electrons because it's protonated. And
Okay.
So maybe it's pulling the electrons away from the carbon ends up being kind of positive.
Yeah, perfect. So nitrogen's formal charge, remember, is just electron counting. It may or may not tell you anything about the actual distribution of charge in the molecule. You can do the calculations, and you would see that the places that are electron poor, that have some positive charge, are the protons and the carbon. And so it's actually the carbon that's towards the ring rather than the nitrogen, which is kind of cool. Cation pi interactions vary in strength in solution, 0 to 5 kilocalories per mole. And so this is another in our list of things that are individually or fairly weak, but overall can contribute a lot.
And you would expect then that if we add sodium hydroxide, then the interactions will be less favorable because
If we add competing cations like sodium, would the interactions be less favorable? Could you compete them off?
more the hydroxide because the base pools.
Oh, yes. So you would predict, at least for some of these, that they would be pH dependent. Yes. Yeah, especially with the ammonium side chains. If it's no longer protonated, then yeah, you should have less of an interaction. I think there was maybe another I wanted to show you. This is in a molecule called acetylcholine esterase, and this is a tetraalkyl ammonium group hovering over a tryptophan. Anyway, that's all we have time for today, so have a great weekend. We'll see you Monday.